Hi, good morning. My name is Li Liu. I'm an assistant professor from ASU, work for the Department of Biomedical Informatics and the Biodesign Institute. I'm very glad to have this opportunity to present our research on computational genomics and how we apply it to identify biomarkers for precision oncology. As a bioinformatician, I always feel that we are living in the golden age of biology because we are presented with new type of data constantly. And since the completion of the Human Genome Project, we now have a catalog of more than 88 million genetic variations in our population. And we're sequencing clinical samples and hundreds of other species that are living on this earth or has lived on this earth. And with the creation of a national electronic medical record system, we all provide the data power that drive the initiative of precision medicine. While precision medicine permeates many diseases, oncology clearly sits at its vanguard. As other diseases, cancers has a universal inter-patient variability. That is what sparks precision medicine. However, cancers are unique and much more complicated because it has other sources of biological heterogeneity contribute that were observed inside a person and even inside a tumor. And the goal of the precision oncology is to understand this heterogeneity and use these characteristics to help improve clinical practice. While precision oncology has many aspects, my team is particularly interested in treatments. We know that some treatment that works best in one group of patients is not necessarily the optimal treatment for other patients. Ideally, if a doctor can foretell a patient's response to a therapy, then doctors can choose the optimal regimen that gives the optimal outcomes. But that requires an accurate predictive model, and that is what our lab is working on. We want to find the biomarkers that can give this accurate prediction. Well, in my opinion, a good biomarker or group of biomarkers first needs to play well the number games. But second, it also needs to have biological implications. And in order to find these biomarkers, I want to first understand how cancer evolved from normal cells into malignant mass. So the story starts with one single normal cell that acquires a mutation. This mutation gives it a little bit growth advantages. When this cell divides, all its descendant cells will carry this mutation and form a subclone. Later on, another cell from this subclone will acquire another mutation, further boost its growth rate. And this becomes a new subclone. That process was repeated several times and eventually grows into a malignant tumor. But when we detect this tumor in clinical setting, we have already passed all these stages. That would mean it has a group of subclones that we need to find drugs to target on. And that is the principle of chemotherapy cocktails. Well, the thing is, sometimes we cannot target all of the subclones simply because we don't know what are there. Okay. If only certain subclones are suppressed, the long-term outcome for a patient is not that optimistic because eventually those subclones that were missed will grow back and reclaim its lost ground. So for us, in order to predict a patient's outcome, we need to reconstruct this path of subclonal evolution. And then we need to do this for every single patient because this roadmap is different for each of us. And later on, we will map the drug target in a therapeutic cocktails onto this roadmap, and then we'll have a better picture of what to expect for this patient under this condition. And another complication in cancer evolution is that not every single mutation in a subclone will deliver that growth advantages. those mutations that they do deliver this advantage drive the formation of the cancer. Others are just passengers. So the question is, how can we tell driver mutations from passenger mutations? The approach we take in our lab is, when we look at a human gene, we don't just constrain us to our own species. We look beyond the human genome to compare this sequence, the sequence of this gene across multiple species in primates, in mammals, and we sometimes even go further to vertebrates. So this is an example. If we have a gene, we would retrieve its sequences for 40 to 100 species, and we compare them. If this sequence remains almost the same across these hundreds of million years of evolution, we would consider this gene has very essential function, because species or organisms that carry a variation of this sequence has been purified from the species pool due to the deleterious impact. On the other hand, if we have a gene, its sequence vary a lot across species, we would consider this gene may not have critical functions. So that is the story and the relationship between sequence conservation and functional importance. And what's interesting about cancer genes is that the oncogenes and tumor suppressor genes are among the most conserved genes in our genome. And those driver mutations, they disrupt the most conserved positions in this gene. On the other hand, for genes that are not associated with cancers, their conservation level is low, and the passenger mutations are observed at highly variable sites. So with this evolutionary information, we go ahead and perform integrative analysis to find biomarkers for precision oncology. Usually, we will start with a huge data set that has clinical information and an array of omics information. Comes from the genome, the transcriptome, the proteomics, and epigenomics. And we will overlay everything with the evolutionary path. Usually, the data set will contain a few hundred patients, and each patient will have millions of features. And because of this high dimensionality of the data set, we will have to use some very powerful computer cluster to crunch these numbers. And we use machine learning method to discover the patterns hidden in this data set. And our goal is to find a small number of biomarkers that has the highest discriminative power to foretell the treatment outcomes. And also, we want these biomarkers to target key evolutionary paths. Okay? And that is how we take big data and translate them into biological knowledge. And here I want to give you an example and talk about a small project. We apply our method to predict the treatment outcomes in acute myeloid leukemia patients, or AML. AML is life-threatening cancer with rapid progression. So identify the optimal therapy as early as possible is critical to patient survival. But the individual variability is very high in AML. Different clinics has reported that the rate of cure can vary from 5% to 40%. And the rate of resistance to drugs can vary from 30% to 90%. Well, doctors are aware of this. So in the WHO or European leukemia treatment guidelines, they clearly stated that you need to classify your patients into one of these 3 risk groups and treat them accordingly. But even with this stratification, within each group, there's still a lot of variabilities that are not tackled and are not solved yet. So in 2014, how can we accurately predict the treatment outcomes for AML was selected as one of the dream challenges. And this organization invites scientists from worldwide to analyze one data set from MD Anderson Cancer Center, and then we compete for the best predictive model. And this data set compared to our to the data set we usually work on is relatively small, but it's very valuable because it's one of the very few proteomics data set with comprehensive clinical data available now. It contains 291 AML patients. For each patient, they measure 40 clinical parameters, and they measure the expression level of 231 proteins. The goal is, can we use these parameters to predict if a patient will develop complete resistance or complete remission to the induction therapy? So the goal is very clear, well-defined, and then we went ahead and took the data set and play our game. After we built the model, we found that we are not given the complete data set. The organization held half the data set to themselves for blind testing. And to our surprise, we won the first place among all 31 groups, highest accuracy close to 80%. And that is 5% higher than the runner-up, and it's 15% than the current standard of care. So we claim that we do improve the prediction quite a lot, but there is still room for improvement. And later on, at the summit meeting, we compared our notes and see how one group developed a model compared to others, and we found computationally, we don't do anything different from others. What is unique about us is the evolutionary patterns. That is what made us stand out. And we didn't stop there. We actually looked into our model and see how many biomarkers we have found, and then are they have any biological implications, because that is the advantages we think of our own model. And indeed, the top two biomarkers both are drug target. The first one is PIK3CA, that is a very well-known drug target in treating leukemia. The second one is GSK3, that was only recently proposed as a potential drug target. And what is important is that when we build a model, we only use evolutionary pattern. We didn't use any drug target information. So this truly demonstrated the power of evolution-informed machine learning in moving forward the precision oncology. If we look back into the history, we will see that doctors has long recognized the challenges of individual variability to clinical practice. That is why they call medicine an art instead of a science. I'm very glad that 100 years later, we are now really moving medicine from an art to a science, and we're part of that. And my team works in the Biodesign Institute, ASU Tempe campus, and also the Department of Biomedical Informatics on the Mayo Clinic campus. And I want to point out that these two facilities, these two organizations, both provide outstanding environments and very valuable opportunities that bring biologists, computer scientists, and clinicians together to work on interdisciplinary projects. So if you're interested in our research or our department, we have tables set up in the other room, and you're welcome to come and visit us. And thank you, and let's keep evolving.